Can you spot the difference between these two groups of tomatoes? To the human eye, they look identical, yet their gene sequences are different. The tomatoes in the bottom row have had 2 genes modified to make them sweeter. In another study, scientists created heat-resistant tomato plants by deleting a gene, increasing their survival rate 3 to 4 times compared to regular tomatoes. These 2 experiments are examples of what gene editing can do. But before we dive into gene editing, let's talk about the grandfather of gene editing, selective breeding. Selective breeding is the traditional method where we find plants with desired traits like size, yield, and disease resistance, and crossbreed them over multiple generations. It works, but it's a slow process, taking 10 to 15 years to develop a new variety. Plus, it's not very precise. Now, let's jump into gene editing. This cutting-edge technology lets scientists change an organism's DNA by adding, removing, or tweaking specific letters in the genetic code. With gene editing, we can introduce new genes that provide an advantage in certain environments, or remove genes to stop the production of proteins that cause disease. In medical treatment, gene editing holds the potential of treating virtually all genetic diseases, including conditions like cancer and neurodegenerative diseases. In agriculture, it can develop crops that are resistant to pests and diseases. And in biotech, it can modify microorganisms so that they are better at breaking down pollutants and toxic substances in the environment. One of the newer tools used in gene editing is CRISPR, which has been a game changer in the field of genetics. Scientists discovered this system in bacteria when they observed that bacteria used a protein called Cas9 to detect and cut viral DNA, effectively disabling the virus. Recognizing the potential of Cas9, researchers adapted Cas9 to cut and modify human genes or other organisms. Here's how CRISPR works. Scientists use a molecule called guide RNA, or gRNA, to direct the Cas9 enzyme to the specific DNA sequence they want to edit. Think of the gRNA as a GPS that guides Cas9 to the target DNA. Once at the target, Cas9 acts like molecular scissors, making a precise cut in the DNA. After the cut, the cell's natural repair mechanisms activate, allowing scientists to disable, insert, or modify a gene. This technology won the Nobel Prize in 2020 and is being further developed to make precise changes in a single DNA strand, or to turn genes on or off without altering the DNA sequence. Now, let's talk about one of the things that matter the most to us. How close is this technology to cure genetic diseases? While the gene editing technology was adopted in 2012, we didn't have to wait that long to hear about the first results of gene editing in humans, though this was extremely controversial. In 2018, a Chinese scientist named He Jiankui edited 3 babies' genomes that had HIV-positive fathers to make them resistant to HIV. He used CRISPR-Cas9 to disable the CCR5 gene, which codes for a receptor that HIV uses to enter cells. Without this receptor, the HIV virus cannot replicate, and the babies are expected to live normal lives. So what was so controversial about this study? Well, while gene editing research is allowed in China under strict regulations, conducting experiments on human embryos with the intent to create live births is strictly prohibited. He Jiankui still went through with the procedure, and after a government investigation confirmed he edited embryos, he received a 3-year prison sentence and was released in 2022. According to He, the children are perfectly healthy. Scientists have analyzed the children's entire gene sequences and found no modifications to the genes other than CCR5. No scientific papers were published because of ethical concerns and problems with how the experiment was designed and carried out. Despite this rogue experiment, gene editing technology has advanced in a regulated manner. In December 2023, the FDA approved the first gene therapy called Casgevy for treating sickle cell disease. This condition causes intense pain due to deformed blood cells that block blood vessels. Researchers used the CRISPR-Cas9 system to disrupt the BCL11A gene in patients' own blood cells, allowing them to produce a different form of hemoglobin, reducing the number of deformed red blood cells. The results are impressive. 96.7% of patients in the study did not experience blockages that led to oxygen deprivation and painful episodes for at least 1 year. Additionally, 100% of the patients remained free from hospitalization during the same period. This breakthrough highlights the potential of gene therapy to transform the treatment of genetic disorders. Casgevy is an ex vivo treatment, meaning scientists remove some of a patient's cells from the body, edit them genetically, and then reintroduce them. The treatment is expensive and requires chemotherapy to destroy faulty stem cells in the bone marrow, ensuring the modified cells can successfully replace the damaged ones. Now, here's where it gets really exciting. Pharma and biotech companies are working on in vivo technology. They aim to inject patients with editing tools, deliver them to the right cells, and edit genes directly within the body. That's so exciting for me. One of these companies working on the in vivo technology is Mammoth Biosciences. Co-founded by Jennifer Doudna, a co-inventor of CRISPR technology and Nobel Prize laureate in chemistry. Mammoth Biosciences uses CRISPR for both diagnostic and therapeutic purposes. They are creating a platform of miniaturized CRISPR, small enough to fit into current delivery systems, ensuring they reach the right cells when injected into the body. I really want to make it clear that a few years ago, I wouldn't have thought this possible. Modifying the genes of a grown-ass adult and effectively curing genetic disorders would be a dream come true for many people. In this scenario, there wouldn't be a need to destroy the diseased cells, as those same cells would be modified to become healthy. Did you know that bacteria can influence our cravings without us even realizing it? Watch this video to learn how you can regain control of your eating habits.